but we were going to cover this topic anyways in lecture. It's just a short slide set. And um, so this idea is not new. This is the idea of taking a part of the plant body and growing it into a brand new plant. It'll be a clone of the mother plant you took the cutting from. And so it's a really nice procedure to do if, you find this perfect plant and you're like, oh, I want more of that. You could take cuttings and grow it, and it should resent as long as you give it the same kind of environmental habitat, it should look like the mother plant. Cuttings are widely done in industry for a variety of reasons. And I was talking to as part of the indoor ag program, there's an advisory council of business people and entrepreneurs that advise. So they're the council. So they advise into our program of what they're looking for students, the training they get. And so we interact with them a lot to get their feedback. And when we were designing the program, they were helping with all of that because they were super excited about this program because they had so much input into it, and they knew that once students graduated out, they would be immediately employable because we listened to what the advisory council had to say and started implementing the concepts they wanted those students to know and skills they wanted those students to know. And one of them was propagating. And one of the council members said, you'd be surprised how people don't understand how to take good cuttings. And so this is a first taste of that. And so you can see that's a potato field. That was one of my research plots. And these are potato tubers that we would plant in there to get them growing. And you can see that they're already producing buds. So those will, if you plant them and tend to them, those will grow into the potato plants. And so certainly that kind of cloning is routinely done. That's how we used to grow potatoes. We didn't start with seeds. We started with the tubers themselves, and that worked really well. And then here you can see they're taking cuttings of the plants in the background and putting them into water, which for the lab tomorrow, I would encourage that if the plant lends itself to that idea, putting it into bottles of water so you can watch the roots, the adventitious roots form, I think is a great idea. And so plants are remarkable that we can cut them up like this. And they're like, yeah, whatever. And this is a great quote from Stefano Mancuso, a scientist in Italy, where he studies plants in detail. But he said, plants distribute over their entire body the functions that animals concentrate in specific organs. Decentralization is the key. We have discovered that plants breathe with their whole body, see with their whole body, feel with their whole body, and evaluate with their whole body. Spreading each function over the entire organism as much as possible is the only way to survive predation and damage. And plants can do it so well that they can even withstand removal of much of their body without losing functionality. So, I mean, it's just amazing what you can do to plants and they just bounce back. And so these cuttings, don't feel bad about it tomorrow. Yes, you are wounding them, but they will bounce back and you'll get a beautiful plant, hopefully, out of this if you can get that plant to form those adventitious roots. And and then you'll eventually put it into soil if you have it into the water first. So stem cuttings are probably one of the most common strategies to use. And so you need to know a little bit back on the anatomy of the plant where you have node regions where you can find the axillary buds. Usually a node region is where you see a leaf or a branch coming off that stem. And so you're hunting for something like that. And then here's a kind of a blow up of the node region. So that region will kick in. So even if you don't have a shoot tip, like if somebody took that portion already and got rid of that, then it's okay because you can take a cutting roller down and that axillary bud will get active. And so you can take multiple cuttings off of one stem if it's long enough, okay? And then typically what you do is you'll often remove the lower leaves so that you can put the stem in deep enough into the water. What was the other thing? So know the idea of the petiole, that terminology, you'll need to know that for tomorrow. So the petiole is like the little stem that leads out to the leaf blade, but we do call it the petiole. I'd like you to know that. Okay, so the petiole and the leaf blade. Some of you may choose to do that. You might cut off where the petiole is attached. Like if there's African violets, that would be one way to do. You just cut off right there and you put that into you probably put that into soil rather than water with African violets. So you take pieces of those tissues and give it the right environment. First, you're going to have to provide it with water because you've definitely gotten rid of its root systems, right? And so if you take a cutting, that cutting has no roots. And so there's a lot going on. That plant, it has to send oxygen down to the cutting, the cut area, and tell those cells stop being stem cells, instead turn into root cells, and then adventitious roots will form. So it's an amazing process. And if you take care of the plant, it will then form a nice root system and continue to grow as long as you continue to care for it. So you can take several cuttings along the stem, and as long as you have an axillary bud, you should be fine. It will grow well. And like I said, often you remove the lower leaves. So there will be some stem cuttings that are going to be options. I'm going to double check that we have some African violets in there. I don't know if we do. If we don't, I might bring in some of my African violets at home are in desperate need of division and repotting. And so you could actually help me with that if you want to take some African violet cuttings. So if there's none in the greenhouse, I think I'm going to bring in my poor plants that I have been neglecting. They desperately need to be pared down a little bit. Okay, so this is an idea here too. So this is a plant related to African violet. And what they did was they took a mature leaf and kind of put the leaf blade into the soil and kept the soil moist. I don't know if you can see that well, but you can see all these little baby leaves popping up right there where the mature leaf got put into the soil. So something like that we can do as well. And that's commonly done in the African violet industry, or this is Streptocarpus. I have one like that at home, but I don't think we can propagate that one because it's not robust enough yet. So you can either do the leaf blade, or you can do the leaf blade and petiole and put the petiole into the soil, or sometimes it's just the leaf blade. And then you'll get a little baby plant coming from that. You have to be patient. You have to these don't grow super, super fast. You have to be patient, let the roots grow well enough. So you have to just keep watering and tending to it. And then if there are any succulents on the cart, these are interesting. So here you can see leaves of some succulents were taken off the mother plant. So these are the mature leaves. And you can see what you tend to do with succulents is you take a leaf and you have to let it, they usually say callus up. So where you've detached it, there's, going to be a wound right where the leaf came off the stem. And they want you to let that kind of callus up a little bit. And so they say just leave it resting on soil or a piece of paper towel for a couple of days. They usually say 2 to 7 days, and then go ahead and plant it. And that, from my understanding, that helps reduce rotting from fungi getting in and causing problems. So, but you can see the little baby plants are coming up from that. This looks like maybe African violet leaves now, and you can see the baby coming in. And then this one on the right is really interesting. What they did was they took a leaf and then they took a scalpel and they wounded it. And you see that central vein? I don't know if you can see where you're sitting, but they made these slashes across the central vein. And when they did that and then gently pressed the leaf onto the soil and watered the soil, you can see the little baby plants are starting to come out of that area. So it's amazing what you can do. And then the middle one, that is the genus name is Sansevieria. Its nickname is snake vine or snake plant. It's also called mother-in-law's tongue because it kind of looks like a tongue, and they say it's a mother-in-law's tongue because the tip of it is very sharp. So sharp tongues, it's a joke about mother-in-laws. So here, what you can do, I think we do have some of those on the cart. And so you can see cuttings taken. So that's all leaf tissue, but there's meristem tissue running the whole length of the leaf. And so here you can see someone came along and took off the tip. So with this one, and Jacqueline will tell you again tomorrow, you need to make sure that when you take a cutting, so this one's like the middle of the leaf, right? It's not the tip area, it's like the middle. So if you take this cutting and you accidentally flip it and put it upside down in the soil, you'll get roots coming out of the top. It's weird. So make sure that you keep the orientation. If you take a middle cutting, make sure you know which way is closer to the tip, which way is closer to the base of the stem. Otherwise, you're going to get weird results. And then this one looks like it was toward the base of the stem, or maybe that was a tip that got put in upside down. That's probably what they did to show. So it looks like a tip there, yeah. So yeah, they accidentally put it upside down. Just be aware of that. The mother-in-law's tongue or Sansevieria is very hardy plant. It's hard to kill. So if the only downside is you have to really be patient. Once you put the cutting into soil, you have to take care of it, and nothing's really going to show up. The ones that I propagate and bring home, they usually don't really show up anything for a few months. So you just have to be really patient. And don't overwater it. It is, what I love about it is it can handle shade conditions. Like I don't have a ton of sunlight coming into my house, and it's fine with that. So if you have not a lot of windows, this is a great option. So those are some ideas of what you'll be doing. And then just be aware that there are other ways of propagating that they've come up with. So tip layering, where you can just put the tip into the soil and it starts to form roots and new shoots. And then you can cut that away. You can see that once that happened, they cut it away and we'll plant that and we'll get it going. And then simple layering where they just bury part of the stem. And the buried stem area goes, oh, I guess we got to form roots. And they start forming roots. So really interesting ways that you can manipulate the plant to get it to produce new plants from the mother plant. Mound layering is a technique that's commonly done, where from one mother plant, you can get several plants from that just by simply cutting it back and then mounding up the soil over the cut branches and then just leaving it alone. And it'll start watering it, of course, caring for it, but it'll start to form new plants off those cut stems, including the roots. And then air layering is a little tricky. One thing I've had issues with is I forget to check on them frequently enough. You have to keep this moist, but basically you take a cut like that all the way around, and then you usually put sphagnum moss and then wrap it, and then just leave it. And make sure that it stays moist. And then you can unpack it and look at it every once in a while, and you'll see some roots coming out. So lots of interesting ways of propagating. Those are fancier. If you want to try some of these, there's, maybe we can find some plants either in the greenhouse or order some where we could try some of this if you want. But tomorrow we're going to start with kind of the basic idea. Okay, any questions on propagation? So once you get this propagating going, it's your plant, like you can take it home. So I wouldn't take it outside anytime soon. Students usually keep it on the grow rack until toward the end of the semester, because if you take them out when it's too cold, you'll shock them. It's shocking. Okay, so that will be fun. And like I said, I'll write a note to myself to bring my poor, sad African violets. What was the other? Okay, so then let's go back to the other slide set. on plant growth. We had one other. So we were looking at mimosa, the sensitive plant, and Venus flytrap, the sundew, and how that is displaying seismonasty, usually some kind of outside mechanical disturbance, an insect crawling across it is the most appropriate thing that the plant wants is an insect so that it can do its action. So those are turgor movements. They're very fast to hopefully trap an insect. All those plants tend to live in well, not the mimosa, but the Venus flytrap and sundew live in nitrogen poor, boggy environments. And so having a luscious insect with lots of nitrogen, just what it wants. Here's the last idea I wanted to talk about, which is thigmomorphogenesis. Big long word. If you dissect that, thigmo means touch. Morpho is its body, the shape of the plant body. And genesis means to create. So this is a shift in morphology. You're creating a morphology through touch. Let's think of it that way. You're creating a kind of a different morphology through touch. And it doesn't have to be a solid touch. It can be wind too. So wind movement can cause this. I've seen that with plants that I purchased, and I put one on my porch in the summer, and I put one in the bathroom that doesn't have any windows and no air movement. And the one on the porch looked like this. Well, I mean, it wasn't this plant, but the one on the porch was more like this, short and stocky. And the one in my bathroom was tall and leggy. And it was just through wind, the action of wind causing that difference. And when you have some kind of physical disturbance, mechanical or physical disturbance, you tend to see those plants get shorter and stockier, their stems thicken up. If you've ever gone on deer paths in the trails in the woods and looked really closely, not now in the winter, but in the summer, there's plenty of deer paths that I've wandered on in the woods, and you'll see that the plants that are flanking the path tend to be shorter and stockier than the same species that's out a little bit, that's not getting constantly brushed against by a human walking on the trail or a deer or a dog or whatever. And so to get this result, they actually touched the plant with their hands twice a day, and the other one they left untouched. So if you do touch your houseplants, you are impacting them. You will create a situation where they tend to stay shorter and stockier. They will respond to that. So thigmomorphogenesis. Okay. All right. So now let's see. So those turgor movements, the sundew, the mimosa, the Venus flytrap, it's all just the turgor pressure that can be quite rapid. If you've ever watched those videos I put up there or have seen Venus flytrap or any of those do their closing of the leaves, it's the pulvini region. And so that's just a special area of tissue where you're going to see that water coming in or out of those cells very rapidly, and it causes the leaves to close up. And so with the sensitive plant, and the sundew and the Venus flytrap, those are all very, very good examples of that. It's a turgor movement that can be very quick. So here's a mock-up of the sensitive plant, and they're trying to show the pulvinous region. And so it's just a stretch of tissue there. And that's where you're going to see the water coming in and out. And it's usually an asymmetrical thing in order to get the leaflets to close up, and then the whole leaf will kind of tilt down. Showing here too is that you get the tilting down of the leaf. That usually cause insects to roll off the leaf and wander on to the next plant. It's another mock-up of that. Okay. We also see it here in a flower. This is really interesting. So this is the stigma portion of the flower that you see here. You can see the three sections of it here. So that's the stigma, and it's on a stalk that you really can't see from this vantage point. But the stigma is on the style, which is then connected to the ovary. And it's the stigma that receives the pollen grains. And so in this case, the stigma is opened up. It's showing the three partitions, so it's in the open position. So in this state, it's ready to accept pollen grains. But then either before it's ready to go, so if it doesn't have its ovary and eggs ready to go, the stigma will be closed up and won't be accepting pollen grains. Also, once it does get pollen grains, it also will close up then too. All right. So it has this ability to, I'm going to close up because I'm not quite ready yet. I got to get my eggs ready to go. Okay. Now my eggs are ready to go. Then it opens, then it gets pollinated, and then it closes. So that's open. Let me show you the closed. So that's closed. You can see it's really closed over on itself. So this is also turgor movements that's causing that because it is fairly rapid. Once it gets pollinated, it tends to close up pretty rapidly. So that's a cool movement there. One other one, Nyctinasty. I forgot about that one. Nyctinasty. These are day-night rhythm movements based on day versus night. And the classic example is the prayer plant. And so these are common houseplants. And during the day, it looks like that top image, figure A. And then at night, it looks like figure B. It looks like the leaves kind of go up and they kind of go like praying hands. I bought one once and when I was teaching this class and I had it at home. And I remember creeping out, I'm sound asleep and I wake up. I'm like, oh, I'm going to check on my plant. And I go and check on it, and it was never in the prayer position. I'm like, I got a mutant plant. I was so upset. But I think what was happening is there was enough street lamp light coming through the curtains where I had it that it wasn't responding because when I brought it to school, I stuck it in a cupboard at night, although I didn't come over here in the dead of night. But it was working because I'd come in and I'd get it out of the cupboard and it was still in the prayer position. So just be aware it might not work as obvious as this if you have it at home, if there's any stray light. And so again, it's a turgor movement. It's not as rapid as Venus flytrap leaves closing, but it is definitely once they sense darkness is descending, they will go ahead and do this. And it tricked me. One year we had a bunch of bean plants in the greenhouse. We were running an experiment, and I went into the greenhouse and it was dark. And so I flipped on the just the task light and I went in there and all my bean plants were doing this. Right. I went into panic mode. Oh, they're wilting. Oh no. This was an experiment. We were going to publish it. And I just went into panic mode. And I went to all the pots and I'm like, no, there's plenty of water. And then it dawned on me, oh, they're doing Nyctinasty response. And so I stopped panicking. But yeah, so during the day, bean plants tend to do, the normal, have their leaf blades out, but at night it's very often, if it's dark enough, they'll go ahead and droop like that. They think it might give them an advantage in really humid conditions to tilt their leaves like that. If you can imagine like a steaming jungle, and if a bunch of water starts to pool up on the leaf blade and just sit there all night long, it's a higher risk for a fungus to start, mold starting to get after the leaves and attack the leaves. So they think this action helps, water droplets roll off and they won't get attacked by fungi as readily. So pretty simple idea, but it can be pretty effective. And then solar tracking. If you've ever driven by a field of sunflowers downstate, I can't remember exactly where I was. I was on the main highway coming back north and there's a beautiful field of sunflowers. I made my sister pull over, like, we gotta go check that out. Anyways, the solar tracking, you'll see sunflower plants. It's very obvious, especially when you have a big, beautiful field like that, because they tend to put their leaves in their big flower head, tracking the sun. And so they can do this through turgor movements through the day. Again, it's not super dramatic. It's, because the sun is moving quite slowly across the sky from sunrise to sunset. But you will see that they do track that. And so you can call it solar tracking. Another fancy word is heliotropism. Helio is sun. The means sun. So typically, a lot of plants will do this heliotropism where their petioles will twist a little bit to try to get their leaf blades, as I said the other day. They want to track, they want to hit, they want the sunlight energy to be hitting their leaf blade at a 90-degree angle. So at high noon, the leaves want to be horizontal so that when the sun is striking the leaf blade, it's 90 degrees. That is optimal for the plant to absorb the most sunlight energy for photosynthesis. And so the petioles can do these turgor movements. And then the other thing too that's really interesting is a lot of plants do this where if you put them, if they're living their life outdoors and they're tracking the sunrise and then the movement through the day and then sunset. So that's what's happening in figure A, B, and C. And then in darkness, they don't stop moving. They'll start to orient themselves toward the east again, waiting for the sun to come up. And so they know that this is temporary darkness and the sun's going to come up. I'm going to get my leaf blade ready to go. So as soon as the sun comes up, we can start doing photosynthesis. So that is something that you can also see. And then this, you may have noticed if you've ever gone walking through a field of like grasses, native grasses after, a pretty good period of drought. You'll often see the leaf blades curl up like that. And sometimes it can also curl up like that due to an insect hanging out in there and creating webbing like silk to tighten up the leaves around its body. But that's not what I'm talking about. This is the leaf doing this, not the insect living inside there. And so the leaf blades along the whole length, along the perimeter of the length of the blade, will roll. And it's just a way to try to conserve more water, really limit their surface area so that they're just trying to hold on to all the water they possibly can that's already in their body. And so very often they'll curl like that. So if you are hiking through an area and you haven't, you don't know the status of, when did it last rain, look for that because it's pretty obvious when you see that. Okay. So as you can see, like with the solar tracking and all of that, plants really do pay attention to light. Obviously, that does make sense when you think about the idea of photosynthesis. And so we've already talked about phototropism a fair bit in this class where they noticed, Charles Darwin and his son noticed plants were bending toward their windows and at the incoming light. And so Charles Darwin didn't do this work, but later, centuries later, they're like, well, is it any quality of the sun? So as you know, you probably in middle or high school, your science teacher probably put white light through a prism and it broke into all the different colors of light. And so those correspond to different wavelengths of light all the way from around 800 nanometers, red or far red, and then red, orange, yellow, green, blue, indigo, violet, as you get into smaller wavelengths of light. And so they were wondering, like, does the plant respond to all of those wavelengths of light, or is it keying in on something? And when they did the experiment with phototropism, what they did was they would just hit the plant with different wavelengths of light, and it really only responded well where it would start bending toward the light in the blue region. And so right around 450. If they hit the plant with that quality of light, that wavelength, then the plant responded. All the other wavelengths, the plant didn't respond to that unidirectional light. It didn't do that phototropism. And so they started hunting for what in the plant is paying attention to blue light. And they found it. They found a phototropin photoreceptor. Okay, it's a protein with it's a photoreceptor. And so this is a little bit more complicated than, what I was telling you initially, because you have a whole process going on inside the plant where the plant has that phototropin photoreceptor. It will absorb that blue light coming from the sun, and then that kicks off a whole signal cascade to tell the plant, go ahead and start growing toward that unidirectional blue light. And it's, as we said before, auxin's part of the signal cascade where auxin shifts over to the dark side of the stem and starts telling those cells, go ahead and divide. So this phototropin, is it really important for that? You have to have a fully functioning phototropin for it to pay attention. The other huge thing is they've noticed the stomatal opening and closing, so those guard cells either moving away from each other and revealing the pore or closing over. All of that is, well, the opening is stimulated by blue light. And so again, when the sun comes up in the morning, it's the blue quality of light that will trigger the stomata, the guard cells, to go ahead and reveal the pore. Here's some of the data in our experiment. It's just showing if you have unidirectional light, and you do different qualities of light. There's blue quality, green, you can see all the other ones except blue. The plant just kind of ignores it and doesn't bend toward that, but when it gets hit with blue. And so it is something that they've been playing around with now, really investigating, okay, so phototropin, are there other photoreceptors that the plant has? And certainly they have found more. We'll talk about another one too in just a second. And so plants really do have to be able to tell if they're in sunlight versus darkness. Are they getting shaded out by another plant by them? They need to know that as well. So they can actually measure the length of the day and the length of the night. And it's really important, especially when the scientists started looking at this idea, they were very, very interested in how does a plant know when to flower? If you've ever walked and hiked around in the springtime versus summer and fall, you know that certain plants will always flower in the spring, others wait until the fall. And the plant is doing that on purpose. It's paying attention to how long am I in the light, how long am I in the dark, and then it will, based on that ratio, it will decide to flower. If they are a short day plant, so we have them categorized, we say they're short day plants unless they won't flower unless the day length is shorter than some critical period. So maybe it has to be less than 10 hours of sunlight. It can vary what the length of time is. So we call those short day plants. And then there's another category we call long day plants. They're not going to flower unless the light period is longer than some critical period. So if it's longer than 10 hours, then it will flower. So if I'm not mistaken, short day plants are cannabis is a short day, right? I believe. Poinsettias, strawberries, those are all examples of short day. Long day, potatoes, wheat, lettuce. So here's a poinsettia, and the one on the right that's still all green and not showing any beautiful flower design got hit with too much sunlight, greater than 10 hours, and it therefore just didn't flower. I think I might have told you about the poor, did I tell you about that? The employee who accidentally turned on the greenhouse light and ruined a whole batch of, so it was like this kind of setup where they were growing poinsettias for the holiday. And the employee went in there at night and turned on the light, and they all stayed green. And so that whole greenhouse was shot, and they couldn't sell any of that in time for Christmas and all of that. I heard this, it was a podcast. And so when the owner went into the greenhouse, this is what he was finding. He didn't see any beautiful poinsettias. And the interviewer said, "Oh, I bet you fired that employee, didn't you?" And the employer said, "No, actually, I kept him on. He'll never make that mistake again." Like, good on you. I mean, can you imagine you're expecting that and you walk in after, a couple weeks and you still see nothing but green. So that's a big loss. So by the way, the fancy red, those aren't flower petals. That's actually modified leaves. We call them bracts, Bracts, bracts. I have a hard time saying that word. And the actual flower is just this little cluster in that right picture. Those are the flowers. It's multiple flowers all clustered together, and then it's surrounded by leaves that have shifted to that beautiful red color. So it's big business, understanding your plant and knowing how to get it to flower when you want it to flower. There's plenty of people who sell cut flowers and poinsettias and things like that. So definitely want to know about your plant. There's intermediate day plants. That means it's got to be just right. Not too short, not too long. It has to be just right. What's that fairy tale? What is it? Goldilocks. Goldilocks. Thank you. Goldilocks and the Three Bears, right? Day neutral plants. That's basically like, yeah, it's pretty not, it doesn't seem to be picky. Just has to be enough light to survive and go through its growth cycle. So day neutral, I have in the notes section, tropical plants, which kind of makes sense because when you live at the equator, you don't get dramatic shifts in how long the day is versus how long the night is. It's kind of even, Steven. So tropical plants, carnations, cotton, roses, tomatoes, plants that are native to more tropical. So this idea of being able to also tell light, how long am I in the light, how long am I in the dark? It also helps them if they live in this part of the world where they have winter looming ahead of them. They need to be able to prepare for that in time. And so it takes a while, especially if you think about some of these big, gorgeous trees, they have a lot to do before the snow hits. And so being able to assess this ratio of light versus dark is super important, not just for flowering, which, humans care a lot about that because we eat so many of the plants that flower and produce fruit. But it's also just important for plants to be able to tell, oh, I better get going and prepare for winter. So these photoreceptors, there's photoreceptors that are assessing all of this light, dark. Do I have to get ready for fall and winter? And so they are absorbing certain qualities of light, and then these photoreceptors, they actually change their shape. So they physically change their shape, and that's enough to start a signal cascade to tell the plant, go ahead and flower or go ahead and start preparing for winter. And so it's, absorbing a certain quality of light, and it's using that as a signal to shift its behavior and decide, okay, I'm going to start making flowers, or I have to get ready for winter, for example. So a photoreceptor is a protein and a non-protein molecule. The fancy name for that non-protein molecule is a chromophore. And it's the chromophore that will absorb the light. And then you have the photoreceptor in total will start to shift its shape. So they actually refer to them as shapeshifters. I'll show you a drawing of this to try to get this across. So these photoreceptors that are being able to tell the ratio of light versus darkness, they've named them phytochromes. Phytochromes. And it's what is allowing the plant to assess light versus dark. It's you don't need to know this, but it's just when you look at it in the lab and work with it, it's just this pale blue protein pigment. And you typically find it in meristematic tissues. So the apical meristem, the axillary meristem, things like that. Actively growing areas. So this is a weird one. So it has two stable forms, and they're both really important. So you have the so-called P sub R form. So P stands for phytochrome, R for red light. So that form of the pigment, when it's in that physical shape, and we call it the P sub R form, it's ready to absorb red light. That's what it's designed to do. It's like, okay, now I can absorb red light. When it does absorb red light, it actually shifts its shape to become what we call the phytochrome FR form of the pigment. And then when it's in the P sub FR form, when it's physically in that form, it's designed to absorb far red light. And if it does that, then it goes back into the P sub R form. So it's like toggling back and forth, depending on when the P sub R absorbs red light, it'll physically shift to the P sub FR form. That one's designed to absorb far red light. Once it does that, it goes back to the P sub R form. So it's really quite interesting. And so they're trying to show that in this mock-up. Okay, so the P sub R, if it gets, it's designed to absorb red light. If it absorbs red light, then it shifts into this physical form that we call the P sub FR. And now in that physical shape, it's ready to absorb far red light. And when it does, it changes its shape back to the P sub R form. We call the P sub FR form the so-called active form because that's the one that will actually go into the nucleus and start interacting with proteins that control gene expression. And so we tend to call that one. So let me show you just a picture here real quick. So here is the P sub R form. You can see, I call it the M form. It looks like the letter M to me. And you can see the chromophore, so that's what's absorbing the red light. So the P sub R form, when it's in the M shape, it is designed to absorb red light. So they're showing that, and then it shifts to the Y, what looks like the letter Y to me, which they call the P sub FR form. And then if you have the P sub FR form in the Y shape and it gets hit with far red, that will shift it over to the M form. And it just keeps, you can play with this experimentally all day long if you want, and you can get this to happen by hitting those forms with the right quality of light to get it from M to Y and Y to M. Except now they call it P sub R to P sub FR. Kind of makes sense? Okay. It's really kind of weird, but it's actually very good because it's a pretty simple procedure here, and yet it tells the plant so much very, very important information. Okay, so think through that a little bit. On Friday, we'll go through this again, and I'll explain a little bit more of how the plant is using this information. Okay?